record.
Okay.
Yes.
I don't know if I can make him co-host, but I'll make sure all the students can record.
Okay, thanks, Jen. Okay, Caleb, whenever you're ready.
Good afternoon, everyone. My name is Caleb Hong. I'm currently a second-year biochemistry major.
And my name is Justin Chao. I'm a second-year biology major.
Justin and I are currently working under Dr. Joseph Genereux in the chemistry department, and today we're going to be talking about the effect of metolachlor on protein structure. So what is protein misfolding? Well, to fully answer this question, we have to look at protein structure to begin with. Proteins adapt a very precise structure meant for specific functions. On the other hand, misfolded proteins have unintended structures that could lead to loss of function or completely different functions. This is obviously very bad for processes that rely heavily on the roles of proteins. So naturally, it asks the question, what causes misfolded proteins? Some common causes include genetic sequence mutations, transcription and translation errors, and environmental stresses, the latter of which we will be exploring. Luckily, our body has a defense mechanism when it comes to misfolded proteins through chaperones. Chaperones identify misfolded proteins and bind onto them. They eventually hand them off to be either refolded or destroyed. Some of the primary roles of a chaperone is to regulate proteostasis, prevent aggregation, and maintain processes that rely on proteins. On the figure on the right, we have an HSP40 chaperone binded to a misfolded client. It eventually hands off the client to be refolded. A larger issue arises, however, when misfolded proteins begin to accumulate and form aggregates. The abundance of aggregates can titrate away chaperones, which again are our defense mechanism. This leaves the entire proteome vulnerable to proteostasis collapse, and proteostasis collapse is the basis for many neurodegenerative diseases today, including Alzheimer's, Parkinson's, and Lou Gehrig's disease.
This leads us to our topic that we'll be discussing today. Metolachlor is a chemical that belongs to the chloroacetanilide family. This family is characterized by the molecular structure on the right. What's notable about this chemical family is that it contains a potent electrophilic moiety. Previous studies on chloroacetanilides has demonstrated that it is capable of reacting with protein side chains through a nucleophilic substitution and can result in misfolding of the protein. When the chloroacetanilide reacts with the protein side chain, it produces a protein adduct when the chloroacetanilide adds onto the side chain of that protein. Because we are modifying the structure of the protein side chain, we therefore alter its structure and therefore compromise its function. This can result in protein misfolding, protein aggregation, and the neurodegenerative diseases discussed previously. On a more macroscopic scale, metolachlor is used widely across the agricultural and farming industries as an herbicide. Because of the possibility for it to cause protein misfolding in human exposure, investigation into metolachlor and its toxicity to humans is imperative.
Given the reasonable concerns associated with metolachlor and the direct exposure to farmer, we need a reliable method to fully investigate the effects of this herbicide. We can begin with cell culture. By transfecting DNA JB8 plasmids into our cells, we can effectively regulate the expression of DNA JB8 proteins. Now, DNA JB8 is a chaperone with the H31Q mutation. Essentially, this means that DNA JB8 will tightly bind onto misfolded proteins, and the mutation means that it will not hand off the misfolded proteins in a later step. We can then follow up with a metolachlor cellular treatment to induce misfolding. This gives us several degrees of freedom when selecting our conditions. For example, we can do a 30-minute treatment at various concentrations of our choice. We can then do an optimized immunoprecipitation assay to isolate our misfolded proteins. After the metolachlor treatment, we can harvest our cells into respective tubes. We can then lyse our cells with RIPA plus protease inhibitor cocktail, which breaks up the cellular components and separates the proteins from the rest of the cell junk. When we centrifuge our tube down, we are left with a supernatant, also known as the lysate, that should be full of proteins. We can quantify and dilute this. With the rest of the lysate, we transfer to the Cephros beads and rotate at 4 degrees Celsius for 30 minutes. The purpose of Cephros beads is to bind to non-specific proteins, as well as any extra cell junk that may have been transferred over. At this stage, we can reserve our inputs, which should be full of DNA JB8 proteins. We can transfer the rest of the supernatants onto our anti-flag M2 beads. And the purpose of these beads is to specifically bind to our DNA JB8 proteins, which recall are holding onto our misfolded proteins. So by rotating at 4 degrees Celsius for roughly 24 hours, we can effectively pull down our DNA JB8. At this stage, we can take our clears, which should have relatively little to no DNA JB8 proteins in solution. Now that our misfolded proteins are on our M2 beads, we need a way to elute them off. And we do so by adding 6 times Langley buffer and boiling these M2 beads for 5 minutes. When we boil our M2 beads, we are recovering our DNA JB8 as well as recovering the misfolded proteins on the DNA JB8. And this leaves us with our eluate solution. If we take a closer look at the bottom diagram, there's the structure of M2 beads, which consist of heavy chain, light chain, and the DNA JB8 binding sites.
For a brief recap, we now have 3 solutions that we've reserved from our extensive immunoprecipitation protocol. First, we have our inputs, which were taken post-rotation on Cephros beads. Because Cephros beads pull down cell junk and nonspecific target proteins, this solution of inputs should theoretically contain still DNA JB8 and other little proteins floating around, but be free of cell junk. Clears, on the other hand, were retrieved post-rotation on M2 beads. As you recall, M2 beads pull down DNA JB8s, so this solution should theoretically be clear of DNA JB8. Eluates are our last solution. These were retrieved post-elution when the M2 beads were boiled to release the DNA JB8s that they were previously binded to. Releasing them back into the solution allows the DNA JB8s to return back into our eluates that were then collected. With these 3 solutions, we can analyze them through a variety of analytical techniques. First, we generate gels using SDS-Page gel electrophoresis and analyze these gels in 2 different ways. We analyze eluates using the silver stain. This visualizes the total proteins among eluates. If we can ensure that total proteins among all our samples are constant, then we can conclude that variance among the DNA JB8 content later in our experiment is truly due to the results of the cellular treatment and not due to variation in total amount of proteins stemming from errors in cell culture or treatment. The Western blot, on the other hand, is used to visualize DNA JB8 content between inputs and clears. This is used as a metric to identify the success, to quantify the success of our immunoprecipitation protocol. Theoretically, our clears should be clear of DNA JB8, and our inputs should contain DNA JB8 if M2 bead pull down was successful. And therefore, the Western blot is used to evaluate the efficacy of M2 pull down and elution. If the results of these 2 tests are satisfactory, then we can continue on to mass spectrometry preparation and run. Mass spectrometry and data analysis is the crux of our experiment and is used to quantify and identify our proteins. First, we need to prepare our samples with isolation, cutting the larger protein chain into individual peptides that are easier to identify. We then process them chemically, allowing them to be easier binds to our TMT 6-plex labels. These are this is a 6 set of isotopic labels that allow the mass spec to identify each sample. We then load them into a liquid chromatography column and run them through the mass spec. In our data analysis, we've filtered to remove contaminant proteins as well as proteins that are unconfidently quantified. To identify peptides, the MS spectra is compared against an existing protein database, and that allows us to filter out decoy proteins, contaminants, immunoglobulins, and keratin contaminants. To quantify the relative amount of peptide from each sample, TMT reporter ions from each sample are analyzed. The final data are then normalized to DNA JVA abundance in each sample to determine the apparent DNA JVA affinity for each identified protein. With this method, we can then conclude the relative amount of misfolding for each protein.
So what does this mean for our overall experiment? Well, the goals is to identify the specific protein targets affected by metolachlor through quantitative mass spectrometry analysis. If successful, we can then begin to explore the interactions and mechanisms of metolachlor and the protein targets. The overall purpose of our experiment is to bring awareness to communities who may be unknowingly at risk due to direct exposure, as well as create a basis for exploring neurodegenerative diseases at a cellular level. Thank you for listening to our presentation. I would like to acknowledge the Genereux Lab for the opportunity to conduct our research, the NIH for supporting us through grants, BioRender for the images used in this presentation, and the UGRS team for allowing us to present our data. Thank you so much.
Hey, thank you, Justin and Caleb. So now we have time for about 3 minutes for questions. You're welcome to put your questions in the chat or unmute yourself and ask your question. Does anybody have questions? Okay. So Justin and Caleb, can you tell us a little bit about, in terms of the research that you've completed so far, is there anything that you discovered that was unexpected for you as you were beginning this?
I think maybe not so insofar as unexpected, but we were led on a few times with some maybe false leads into possible proteins that could be more in favor of misfolding due to metolachlor treatment. I think Caleb might have more to say about this, specifically with a protein TDP43.
Yes, so we were initially investigating TDP43 as a result of one of the mass spectrometry runs. After further investigation, it showed no correlation to metolachlor, and I think the general thing that we learned from that was the importance of replicates in our data to show significance in our data, essentially.
Okay, thank you. Any questions from the audience?
Suzanne, I put one in the chat.
Okay, thanks, Rich. So Dr. Cardullo had the question, what is the EC50 for metolachlor? How does this impact your investigations on protein folding?
Can you clarify what you mean by EC50?
Feel free, Dr. Cardullo, to unmute yourself if you'd like to.
Sorry about that.
Yeah.
So it's EC50 is the effective concentration, which gives you a 50% response. In this case, the 50% toxicity level. And I'm asking that because how does that affect your conditions that you're testing protein folding disruption by? Are you within the range in which it is toxic or not?
So as of right now, we haven't determined an exact concentration where the metolachlor becomes toxic. And so as we're running these experiments, we're testing concentrations as high as 1 millimolar concentrations of metolachlor and going down all the way to 500 nanomolar concentrations. So we essentially want to see what happens at each concentration and if there is a significant impact on protein misfolding at the lower concentrations of metolachlor.
Yeah, and to add on to what Caleb was saying, this sort of goes with our the goal of our research. We know that metolachlor has potential to misfold proteins, but we want to ensure that these proteins are ones that are significant in causing disease, in being toxic, and we want to identify the point, the threshold where maybe metolachlor exposure can actually become toxic.
Great, thank you.
Okay, thank you, everybody. So we're going to go ahead and move on to our second presentation. Thank you both, Justin and Caleb.
Thank you so much.
Sure, thank you. Jaden, are you